Welcome to the United Leukodystrophy presents Leukodystrophy 101 with Dr. Watkins from Kennedy Krieger Institute.
Hello, everybody, and welcome to the first virtual ULF conference. Wish I could be in Chicago to see everybody, but in these crazy times, we're just going to make the best of what we've got and hope this works. So, Leukodystrophy 101. Basically, what we're going to do is the basic biology behind the leukodystrophies. So let's do a couple of definitions first. So what's leukodystrophy? Well, if you break down the word, the etymology is from Greek, leukos meaning white, dys meaning ill, and troph, and my pronunciation I'm sure is terrible, meaning nourishment. So what we have is ill nourishment of white, which refers to the white matter of the brain. So if you can do a couple of dictionary definitions, here's a couple. Any of several inherited diseases characterized by progressive degeneration of myelin in the brain, spinal cord, and peripheral nerves. They also say degeneration of the white matter of the brain characterized by demyelination and glial reaction, probably related to defects of lipid metabolism. And I'm going to go on and read a couple more descriptions of leukodystrophy, and then we'll put together some common themes and look at the individual components. So here's from the ULF website. The leukodystrophies are a group of rare genetic disorders that affect the central nervous system by disrupting the growth or maintenance of the myelin sheath that insulates nerve cells. These disorders are progressive, meaning that they tend to get worse throughout the life of the patient. And finally, from the National Institutes of Neurological Diseases and Stroke website, leukodystrophy refers to progressive degeneration of the white matter of the brain due to imperfect growth or development of the myelin sheath, the fatty covering that acts as an insulator around nerve fiber. Myelin, which lends its color to the white matter of the brain, is a complex substance made up of at least 10 different chemicals. The leukodystrophies are a group of disorders that are caused by genetic defects in how myelin produces or metabolizes these chemicals. Each of the leukodystrophies is a result of a defect in the gene that controls one and only one of the chemicals. So if you take all of that and see what you can get out as common themes, we have the brain and central nervous system. We have white matter, myelin or myelin sheath, and demyelination. We have inherited or genetic, and we have growth or development of myelin. We have lipids and lipid metabolism, and progressive and degeneration. So what we're going to do here is we're going to take not all of these, well, I think, yes, we probably will do all of these, and just look at a little more depth for some of these things. So brain and the nervous system. So these are cartoons of, on the left, the central nervous system, and on the right, the peripheral nervous system. So the central nervous system, we mean the brain and the spinal cord. Whereas the peripheral nervous system, we mean all the nerves that come out of the spinal cord and go to all your hands and feet and all your muscles, go to all your internal organs, and control all your movements and bodily functions. So if you take a look at a brain, sorry about that. If you look at the brain, and these would be autopsy specimens of brain, whole brain. On the top, we have, this is a side view of the brain. You have the cortex, which has the 4 lobes, the frontal lobe, the parietal lobe, the occipital lobe in the back, and the temporal lobe coming down here at an angle, and then the cerebellum here, and this is the brainstem, and you can see a little bit of the spinal cord coming down. And then if you look at the brain from a top-down view, Again, this is the front and this is the back, but you can see it's divided in the midline. So if you take a slice through the human brain, and here's a little cartoon on the bottom to show where we took the slice, you can see that the white matter does in fact look white, and the gray matter has a more of a pinkish brown color to it. But you can see the white matter clearly does look white in the brain. And I just want to point out that here and down here are actually open spots. This will become more apparent when we get to looking at some MRIs. But these are called the ventricles of the brain. These are where the cerebrospinal fluid flows through the brain. It percolates from the spinal cord to the brain and back again. But that's what these little areas here and here and here are. So again, we're going to look at some brain sections from different views. And what I want to do here is to show you what it looks like first with just a slice. And this one is now sliced from top to bottom. You can envision that you're looking in the middle of the brain. And again, here are these ventricles. But you can see the white matter nicely here. This is a section, a horizontal section again through the brain. You can't see the white. The white doesn't look quite as white, probably because this is an older specimen. You can see it's encased in plexiglass. It's probably preserved in an anatomy lab somewhere. But you get the idea. But now if you can use a stain to stain the white matter so that you can actually see it much more clearly on the vertical slice and the horizontal slice. How about an MRI looking at the same things? Well, on a normal MRI, the white matter doesn't look white at all. As a matter of fact, it looks pretty dark, pretty grayish. These are the ventricles here. The white stuff you're seeing here are the ventricles, and the white stuff you're seeing around the edge is kind of outside of the brain itself, or the surface of the brain, and not the interior white matter. So these are the kinds of images that this is the one you're probably more used to seeing if you have looked at MRIs. But this is a normal brain. And so what's the brain made up of? So it's made up of billions and billions of cells. And although there are a lot of different types of cells in the brain, there are four main ones. The neurons, or nerve cells, these are the ones that conduct electrical impulses and connect the brain to the muscles and the heart and the lungs and etc. There are oligodendrocytes, and these are the cells that actually make the myelin, that make the white matter that surrounds the axon. And what the myelin does is, as you'll see in some pictures in a little bit, it actually wraps around the wire, the axon of the nerve cell, and acts like an insulator, so we don't get short circuits. Another type of cell that's very prevalent in the brain are called astrocytes. And the astrocytes help to make up the blood-brain barrier. So the blood-brain barrier refers to the fact that the blood vessels, the small blood vessels that go through the brain and deliver nutrients to the brain, don't deliver bad stuff to the brain. We have a way of trying to keep out a lot of the stuff that would not be good for the brain to take up, but you want the brain to take up nutrients like sugar and protein and fats. And that's what is another main function of astrocytes. They take up nutrients from the blood supply and supply them to the neurons and the oligodendrocytes. So you'll see that better in a cartoon in a couple of minutes. And then there's the microglia, a fourth important cell type in the brain. And these are the immune cells of the brain. So here's a neuron. And as we said, the neuron is responsible for the conduction of electrical impulses. So there is the cell body that has the nucleus in it. And there's this long process coming out, which is the axon. This is, and the arrow shows the direction of electrical conduction. So in this case, the axon is connecting to another cell, and the area where it connects is called the synapse. And this may be the dendrites, these little processes from another neuron, like you can see axons from another neuron connecting to this one on the dendrites of the cell body. Or it could be a muscle cell, or it could be a sweat gland, or it could be anything connecting to, that helps connect to your functional organs. And here's a cartoon version of the neuron. Here again is the cell body and the axon, but you see now we've got the myelin in there. So in this case, the myelin is wrapping around the axon. And we'll see this a little more clearly in a couple of minutes. And here's a bunch of dendrites. But again, the conduction of electrical impulses starts in the cell body and travels down the axon, where there are synapses between this axon and either other neurons or other cell types. How about oligodendrocytes? So these are the ones that make or synthesize myelin. And so you can see the cell body of the oligodendrocyte is here, but you see the membrane of the oligodendrocyte makes these big sheets that wrap around the axon of the nerve. This comes from a French cartoon, so we have axon and myelin here. And here's another cartoon of an oligodendrocyte, again showing the projections of the membrane of the oligodendrocyte wrapping around nerve axons to help insulate them. Here you see that on both cartoons, you see these things called nodes of Ranvier. They are little areas where there is no myelin. So there's not a complete contiguous myelin structure that goes all the way down the wire. So in that case, it's a little bit unlike if you're thinking about the electrical wires in your house, that if you had little breaks in them, you would not be happy because you'd get shocked. But it turns out that having these little breaks in the myelin sheath are really important for accelerating the conduction of electrical impulses, which are really mainly ions, like sodium or potassium or chlorine. These ions are what's responsible for the conduction of the electrical current down the wire, down the axon. How about astrocytes? So astrocytes, they're named because they look like stars. They have multiple projections coming out of them, a lot of which end up on the little blood vessels or capillaries that percolate through the brain. And basically this capillary would be completely covered, and I think we'll show this in another slide, would be completely covered with astrocytes to create this blood-brain barrier function, which keeps the bad stuff out. But the important thing about the astrocytes is that they're the ones that connect to the nutrient supply of the blood vessels. The neurons connect to the astrocytes, but not directly to the blood vessels. And here's a nice fluorescent colored micrograph of astrocytes showing they're very pretty cells. They have lots of these projections, and they are a bit star-like. And finally, the fourth type of cell in the brain is the microglia. And these act as like the white blood cells do in your lymphocytes, in your the rest of your body. They detect foreign invaders and react to such foreign invaders. In this picture, the astrocytes are green fluorescing, and the red fluorescing cells are the microglia. And so here's one cartoon of all of them in the same context. They're all in the same picture. And so if you just take a little, and you can see where it's taking a little snapshot of, is on the edge of one of these ventricles, or place where the cerebrospinal fluid flows down here. And so there's another cell type here, which we won't really talk about. They just line the ventricles. But here is where what we want to talk about. Here's the blood vessel. Here's the astrocyte with its little feet all over it, connecting to a neuron whose axon here is covered in myelin, which is being made by these oligodendrocytes. And then around all through the brain, you also have these microglia, which are just acting as like security guards in the brain, trying to look out for any kind of foreign invader, like a virus or a bacteria or even a bad chemical or something that's not natural that it can detect and help to get rid of. And here's another one, a similar thing, but this is from the spinal cord, showing that it's very similar in the spinal cord. The only big difference between the spinal cord cells and the brain cells is that the instead of oligodendrocytes, and I think it shows it here, I'm looking, but I don't see the name, the myelin producing cells actually, it is the same in the spinal cord. My apologies. I'm jumping ahead of myself and talking about the peripheral nervous system. So very similar in the spinal cord and the brain. And so let's look a little closer at myelin, because that's the thing we really care about in terms of the leukodystrophies. The myelin sheath, here's a little better cross-sectional showing that's multiple wrappings around the axon. Oops. And here is an electron microscope view of that. And you can actually see the number of whorls of myelin around an axon. This is the axon here. This is the myelin surrounding it. And you can probably count about 8 or 9 layers of myelin that's wrapped around in a spiral around the axon. Here's another picture to make one point, in that adults have a lot of myelin, but myelin is very, very sparse in newborns. Myelin development just begins in the 3rd trimester of pregnancy, but it rapidly accelerates after birth. So in the newborn, you see a lot of these axons here that have very little, if any, visible myelin on it, whereas an adult has quite a bit. And so myelin is made by these oligodendrocytes, and here's kind of it looks kind of like a window shade that has been unfurled, and that's kind of what happens with the myelin sheath. This is part of the membrane of this oligodendrocyte, and here's the axon, and then this just wraps up like a rolled up window shade around the axon and forms this tight layer to insulate it. So a couple of functions of myelin, as we've been talking said several times, the insulation around the axons prevents short circuits, but it also actually increases the speed of conduction of the electrical impulses. So a lot of times with electrical impulses, they kind of, the further you get from the source of the signal, the lower the signal is, becomes as a function of distance. But the myelin, and especially these nodes of Ranvier that have little places of no myelin, really help to speed along the conduction. So what's myelin made of? Well, it's about 20% protein and about 80% fat or lipid. And so we'll talk briefly about both of those. So the myelin proteins, the main ones are called myelin basic protein, myelin associated glycoprotein or MAG, myelin oligodendrocyte glycoprotein or MOG, and then proteolipid protein or PLP. And we'll get to that in the next slide. Myelin lipids include things with very long names like galactocerebrosides and galactosylceramide and sulfatide and sphingomyelin. And these are the fatty substances that really help give myelin its insulation properties, because as you see, it's about 80% fat. So myelin proteins, just to show you where they are in relationship to the neuron. So here's the neuron, the axon here. And then, and this is the membrane of the neuron. And then you start to see the myelin in this yellowish color here on this. You see that MAG and MOG are pretty much on the surface of the myelin, whereas MBP, myelin basic protein, is in the inner layer of the myelin. And this PLP, proteolipid protein, actually helps to hold the wrappings tight. There's an interaction between this PLP on this layer of myelin and then the PLP on this layer of myelin, and there would be more and more as you got more and more layers. But this is where the proteins are. And what about the lipids? So lipids, as we said, are fats. And what makes a fat fat? Well, long chains of carbon atoms and hydrogen atoms is what makes up fat. And here's just a cartoon of a chemical model of just so the black ones here are carbon atoms. So believe it or not, there are, I think, 16 of these in here if you look carefully, surrounded by hydrogen atoms, which are the white ones. Because there are no positive or negative charges associated with this fatty kind of structure, that's what makes these things good insulators. Now, there are other properties of myelin lipids. They're very complicated chemicals, and I won't bore you with their structures, but just to kind of show you where things are in terms of the lipids. Well, we'll get to, I'm sorry, I guess we'll get to that in a second. Let me see. Yeah, okay, we'll come back to that in a couple slides. But as we said in leukodystrophy, there is a problem with myelination, particularly in the leukodystrophies, is demyelination, where there was myelin and then it gets destroyed, as opposed to another term called dysmyelination, where the production of myelin is not good from the start. So a lot of the leukodystrophies are demyelinating diseases. And you can see in this bottom picture, well here you see a lot of myelin surrounding this axon, and you see these are these nodes of Ranvier, and here's a schematic of demyelination. And as the myelin disappears, it really causes degeneration and progression of disease. I'm sorry. This is remyelination. My screen is so small I can't see the words. But this would be remyelination, where which is a goal that we're trying to achieve with therapy of leukodystrophies. You're trying to get that myelin back. Whereas if it progresses, more and more the myelin breaks down, and eventually the nerve axon is not going to be working optimally. And so here's a microscopic view of a bunch of nerve fibers, each with myelin around them. And this is demyelination, where basically all you see are the axons, but not much in the way of myelin. And then if remyelination could be achieved, it might look like this. It might not be quite as dense and quite as good as this, but it's not bad. So this is the kind of goal of a lot of therapies of leukodystrophy, is to get to be remyelinated. So as we said, leukodystrophy is demyelination. Here's a picture of normal nerves and demyelinating nerves. And as you can see, there may be some myelin left some places, but for the most part, it's not looking pretty good in this picture. If you look at the whole brain section, and I apologize, these are the best sections I could find on the internet, but there's a stain called Luxol Fast Blue that stains myelin a blue color. And you can see in a normal brain, the white matter stains this pretty blue color. And in leukodystrophy, you can see that here you see a little bit of blue stain here, and I don't know if that's even real myelin or not. It looks like just artifact here, but this should all be blue. All this white matter should be this pretty bright blue. So in leukodystrophy, we have a loss of the myelin. So what is the etiology of this lack of myelin? So the possibilities include genetics. There could be an environmental cause, like a toxic cause. Could be infectious, like a virus, like Zika, or what's the other one that was really, it's not coming to me, but a viral infection. Could be an immune system dysfunction, like we see in multiple sclerosis. The leukodystrophies that we are most interested in discussing here are the ones that are of a genetic cause, the ones that are hereditary. So that's what we're going to be talking about here. And by genetic cause, we mean there's a defective gene somewhere. Means it's inherited from your parents. Could be a new mutation. Most often than not, it's inherited. But there's always the possibility of the bad luck of having a new spontaneous mutation. So let's just briefly go over modes of inheritance. Some leukodystrophies have a recessive mode of inheritance, which means autosomal recessive. We'll explore that in a moment. It could be X chromosome linked, where the gene is carried on the X chromosome. Or it could be a dominant trait, or an autosomal dominant. And if anybody listened to the talk earlier on autosomal dominant leukodystrophy, that is one example of the latter. I'm going to focus mainly on the first two, just in terms of reviewing how they're transmitted. We know that we have 22 pairs of autosomes, the pairs are numbered 1 through 22, and then we have the sex chromosomes, either the X chromosome, or where a woman will have 2 X chromosomes, and a man will have an X and a Y chromosome. And so most leukodystrophies are inherited in an autosomal recessive manner, meaning that you need 2 copies of the defective gene to be to manifest symptoms. In this case, both mom and dad carry one normal gene and one mutated gene. And the green is the normal gene and the pinkish is the mutated gene. And so when they pass on their chromosomes to their kids, there are 4 possibilities. Dad can give if dad gives his good gene, his good chromosomes to his kids, it can be this guy here or this child here. So you can have dad passing on, it doesn't matter, one of the green ones. And if mom passes on one of the green ones to the same child, they both have normal, non-mutated gene. These 2 kids, one got their good gene from the mom or dad and the defective gene from either mom or dad, doesn't matter which. Those are going to be just like mom and dad and be carriers of this recessive trait. But if both mom and dad pass on the recessive gene, the gene with the mutation in it, there is a child with the disease, and in this case, a leukodystrophy. So this is the way that most leukodystrophies are inherited. Some, for example, X-linked adrenoleukodystrophy, are inherited in an X chromosome linked manner. So in this case, the defective gene or the mutated gene is on an X chromosome. And mom, in this illustration, has a mutation in one of her X chromosomes and a normal X chromosome. Dad has a normal X chromosome and a normal Y chromosome. And so if dad passes on his Y chromosome to a child, again, here's the two boys, so they will be getting the Y chromosome from dad, and they will obviously be getting an X chromosome from mom. And in this case, the child received the normal X chromosome from mom, and in this case, the boy received the X chromosome with the mutation. And so this child, because he has a mutation in the X chromosome, but instead of another X that has a good gene on it, he gets a Y chromosome because he's a male. Unfortunately, he will be affected with leukodystrophy. If both mom and dad pass on the good X chromosome to a female child, she will be normal. But if dad passes on his his X chromosome and mom passes on her mutation X chromosome, she, like mom, will be a carrier. And while she may not show symptoms, she will be able to pass on this recessive trait to her offspring. So we often get asked, how rare or common is leukodystrophy? Well, as it turns out that some leukodystrophies aren't all that rare. 1 in 17,000 people have a mutation in the gene that causes X-linked adrenoleukodystrophy. And in people of Ashkenazi Jewish ancestry, 1 in 40 people have a mutation in the disease that causes Canavan disease. So they can be rare, but they can be, in certain populations, more frequent. Although many leukodystrophies seem rare, is this due to infrequent gene mutation or underdiagnosis? And that's something that we're all working on. The overall incidence, as best I can get it from the medical literature, for all leukodystrophies is about 1 in 7,500 births That would be worldwide. So when do we suspect that a child or an adult has a leukodystrophy? Often there's a gradual decline, particularly in an infant or child who previously appeared well. So a child is born and develops normally for a couple of years and then all of a sudden starts regressing. That is one sign of a leukodystrophy. Progressive loss in body tone particularly in an infant, does the infant become floppy, not be able to hold his head up as well? Progressive loss of movement or change in gait if the child is walking, speech, the ability to eat, vision, hearing, and behavior. Any changes in these can be signs of leukodystrophy. Slowdown in mental and physical development. The child was developing normally and then all of a sudden it just doesn't seem to be making progress. This could be a sign of leukodystrophy. But symptoms vary according to the specific leukodystrophy. Unfortunately, there is not one size fits all diagnosis of leukodystrophy. And importantly, it may be difficult to recognize in the early stages of the disease because it may just not be a significant enough change to raise your eyebrows. Sometimes only in retrospect do you realize that, oh, when we started not walking as fast or or became more clumsy, maybe this was a sign that there was something going on that we just overlooked as, maybe there's just some clumsiness there. So it may be very difficult to recognize in the early stages. And for adolescents and adults, the rate of disease progression can be slower and less severe. And movement disorders, seizures, and dementia are also common. So how do we diagnose leukodystrophy? Well, the history of the illness, based on the types of issues that we mentioned in the previous slide, are very important because very often there are patterns that will suggest to the neurologist that it may be this leukodystrophy versus that leukodystrophy. Neurological examination is obviously a very important part of the diagnostic process. We also would look for the presence or absence of non-neurologic symptoms because a defective gene may be causing a leukodystrophy, but it may be causing problems in the liver or another organ in addition to the brain. The family history is important, especially the family ethnic background, as that may also provide some clues or rule in or rule out potential diagnosis. But probably the most important thing in trying to diagnose a leukodystrophy is the brain MRI. And here is, and we've seen this earlier, normal brain MRI. As you can see, the white matter does not appear white. It appears darker than the gray matter, which does appear gray. And you see a little bits of white, but these are normal. These are the ventricles again, and these are more surface areas of the brain. But in leukodystrophy, there's often very obvious changes. So all of the white, most of the white that you're seeing here is abnormal and is evidence of a leukodystrophy. Keep in mind that early MRI changes are often very subtle. And of course, like the progression of the disease, the MRI abnormalities generally worsen as the disease progresses. Patterns. Leukodystrophies often have unique MRI appearances, and pattern recognition can often suggest a diagnosis. There are clearly differences in the appearance of XALD and Krabbe and LBSL, and these are a couple other examples just to show that the pattern can sometimes help the neurologist in making a diagnosis. Biochemically, we can diagnose leukodystrophies or help diagnose leukodystrophies by laboratory tests. So these are used along with clinical findings to confirm or refute a diagnosis. Tests are usually specific for a single leukodystrophy or perhaps a group of related leukodystrophies. There are panels or groups of tests for the most common leukodystrophies, and these can be useful because if you can say that, yes, we do this group of tests and we can tell that it's most likely leukodystrophy X. But no single test can answer the question, does my child or a patient have a leukodystrophy or not? So the ULF website lists 56 clinically distinguishable leukodystrophies. But if you look in the medical literature, it suggests that there are probably more than 100 different leukodystrophies. And so genetic defects in leukodystrophy can result in biochemical abnormalities, such as you could accumulate a toxic substance. Toxin to the brain doesn't have to come from the bloodstream. It can be something that the brain actually makes inappropriately. It could be a mutation in a specific brain protein, or too much of a specific protein. The mutation in a specific brain protein sometimes causes the protein not to work. Sometimes it causes it to overwork and make too much of whatever it is that it makes. And so some examples of accumulation of a toxic substance include X-linked adrenoleukodystrophy, where very long chain fatty acids accumulate, MLD, where a lipid called sulfatide accumulates, Krabbe with galactosylceramide, and in Canavan disease, a substance called N-acetyl aspartate accumulates. And usually these substances are normally present, but there's got to be a balance between just enough, too little, and too much. It's the Goldilocks situation. And so you can use this little seesaw cartoon to see what is in balance and what's too much and what's too little. So usually the brain makes and breaks down substances that could be toxic if there's too much of. And sometimes we take these in from the diet also, and sometimes we excrete them in either the urine or the feces or sweat or elsewhere. So when you have something that's toxic, it's usually either you make too much, where you have, excuse me, let's do that first. You make too much, where synthesis of the substance is is too much, and you get too much here. Or if you can't get rid of the excess to balance out the seesaw. So if the breakdown is not working properly, you also get too much. And this is the case in X-linked adrenoleukodystrophy. And here we're showing a cartoon of the brain cells again. And so the very long chain fatty acids that accumulate in adrenoleukodystrophy and the galactosylceramide and the sulfatide that accumulate in other leukodystrophies accumulate in the myelin. Interestingly, the N-acetyl aspartate that accumulates in Canavan disease is in the cell body of the oligodendrocyte, not in the actual membranous myelin sheath that is part of the oligodendrocyte. And sometimes these microglia, which are these little cells here stained in this silvery stain, you have inflammation. The toxic substance in the case of adrenoleukodystrophy is very long chain fatty acids, can cause inflammation. And the inflammation can cause the white blood cells to accumulate in the little blood vessels of the brain, but also you get a proliferation of these microglia in the rest of the brain to try to combat this toxic substance. And so these inflammation, as you probably know, is thought to be a very important component of the bad stuff that happens in COVID-19. If you have a lot of inflammation, that can actually help to destroy the tissue, and in this case would be the myelin. So biochemical causes of leukodystrophy, you can have a mutation in a specific brain protein. For example, in Alexander disease, it's something called glial fibrillary acidic protein, or GFAP. And in Pelizaeus-Merzbacher disease, you have myelin proteolipid protein is defective. And so here you see that the PLP is in the myelin, whereas the GFAP is in the astrocytes. So different diseases cause different abnormalities, but they all at the end cause white matter changes of the brain. And so to achieve a genetic diagnosis of leukodystrophy, we would do DNA sequencing. What that means is to establish the order of the 4 chemicals that make up DNA, A, C, G, and T, to establish the order of them in the patient's DNA. And there can be specific gene sequencing. In other words, if you think that this patient has X-linked adrenoleukodystrophy, you might want to just sequence the ALD gene and see if there's a mutation there. There's also exome sequencing, which means the sequencing only of the pieces of DNA that contain the code or blueprint for making the proteins. There's a lot of DNA that is not make or have instructions for making proteins, or non-coding DNA that we used to refer to as junk DNA, but we now know is very important and maybe disease-causing as well. But exome sequencing is often used when we think that the gene with a mutation makes a protein. Or you can have genome sequencing the whole kit kat and the caboodle of it, total DNA sequence. And of course, why would we not just go ahead and do genome sequencing? Because that'll hopefully give an answer, even if what you thought initially was wrong. Well, it's partly a matter of cost. Genome sequencing is getting more affordable. I've seen places where they're advertising for about $1,000 to do total genome sequencing, and more like $250 for exome sequencing, and perhaps 100 or thereabouts, 150 for specific gene sequencing. But total genome sequencing will generally answer the question, is there a mutation in anything? And that can be very helpful. Recent publications indicate that a combination of clinical findings, the MRI pattern, and genome sequencing can dramatically shorten the time from initial symptoms to a definitive diagnosis. And this is really critical, because if you can learn what the specific diagnosis is, you may be able to target either therapy as they're becoming available, or other treatments, or standard of care. This is a really important thing to try and do, and try to get the diagnosis as soon as possible. And Dr. Vanderver, who was originally scheduled to give this talk, recently put out a paper that had this really nice algorithm for a good way to think about getting to a diagnosis of leukodystrophy. So if you start with an abnormal MRI and you follow the solid arrows are a yes, and the dashed arrows are a no. So if there's an abnormal MRI, and yes, there's a likely genetic etiology. If it's not a likely genetic etiology, that means it's acquired, like an infection or trauma, and you would treat that totally differently, because that's probably not going to be a progressive disease. That's probably going to be something that's transient, whereas a leukodystrophy is progressive. And so is there a recognizable MRI pattern? If no, then you would go to other tests. But if yes, you would pursue confirmatory laboratory testing as appropriate, and then you get a confirmed diagnosis, and then treat accordingly to standards of care and return the results to the family with appropriate counseling. If it's a non-recognizable MRI pattern, you might do at the same time, parallel, measure some panels of biochemical tests that might look for the most common, or pursue the whole genome sequencing if it's available. And eventually the goal is to get to a good diagnosis, and then the family can have the best information as to how to proceed. And if all of these unfortunately fail, then the best we can do right now, besides to pursue it, to be diligent and pursue it more carefully, refer the affected family to rare disease advocacy and research programs, particularly organizations like the ULF and some of the other organizations that you've probably heard about today. So we're coming down to the wire here. I just want to say something about approaches to treatment. So the Holy Grail is remyelination and restoration of normal brain function. That's what we really want to achieve. We want to cure these diseases. Unfortunately, most leukodystrophies do not have a cure. Possible therapeutic options include enzyme replacement therapy, which works for some of the lysosomal storage diseases, where if the brain is making a defective protein, you just IV or into the brain give the missing enzyme and hope that it gets where it's supposed to go and works. It does work for some, but it is really very selective leukodystrophies in which this works. Bone marrow transplantation, also called hematopoietic stem cell transplant, has been shown to be beneficial in several leukodystrophies, and there are several transplant centers in the US and worldwide that are experts in this. And again, for some leukodystrophies, this is now the standard of care. Neural stem cell transplantation can work. It's a good possibility, but this involves stem cells, along with all the ethical issues surrounding stem cell utilization for therapeutics. Gene therapy shows a lot of promise. There are several forms of gene therapy. There's lentiviral therapy. There's adeno-associated virus therapy. And then there are a couple others that are not associated with viral delivery of the good gene. There's something called antisense oligonucleotide therapy and CRISPR-Cas gene editing, which a lot of scientists think that this has the best chance for treating a lot of diseases, including leukodystrophies, because you can actually edit the bad piece of the gene out and replace it with a good piece. There are all kinds of technical issues associated with all of these that scientists and clinicians are working very hard to try and solve the technical issues. And there are clinical trials. I would suggest anybody has a particular question about therapies for any of the leukodystrophies to go to clinicaltrials.gov. Just as it said, just clinical trials, all one word, .gov, and then search for your leukodystrophy of interest and see if there's any clinical trials going on that involve any of these issues. And I don't want to neglect the role of supportive care. Sometimes that's all we can offer, but it's critical, even with any other kind of therapy, to provide the most supportive care that we can for our children and adult patients. So in summary, leukodystrophy refers to a group of diseases affecting the white matter of the brain. White matter is predominantly myelin, the fatty insulation around the wires. Degeneration of white matter is due to problems with growth or development of myelin. Genetic causes are the ones that we are most referring to when we talk about leukodystrophy. These diseases are progressive. Some still remain undiagnosed, although we're getting better. Identifying causes of undiagnosed leukodystrophies is getting faster, which is critical. To the earlier you can treat something, the less damage hopefully it'll cause. And finally, progress towards developing effective therapies for some leukodystrophies is now encouraging. So with that, I'm going to end. And if there are any questions, we only have a few minutes left. But I hope that this has provided you with a broad overview, at least, of what leukodystrophy is. And my email is on the title slide. So if you go back to the beginning of this, if you have any questions that I can answer, I'd be more than happy to. Keely is telling, or Tara is telling me that right now, no questions. So if anybody does, okay, Tara says no questions. So we'll hang it out for one more minute. And if there's still no questions, I will wish you a wonderful day. I hope that you learn a lot from this conference. And I hope sincerely that we can all get together in person this time next year. So, no questions. I will wish you all a good day. Okay, thanks, Tara. Take care.